on His Majesty's service. Receptors, Part 2. Merkel discs or Merkel cell neurite complexes are located in the epidermis. They are especially dense in the fingertips, lips, and external genitalia. Accumulation of Merkel discs in touch-sensitive skin areas is called touch domes. Merkel cells are oval and 10 to 15 micrometers in length. They can be differentiated from the surrounding epidermal cells by their large, multi-lobed nucleus. Numerous dense core vesicles measuring 70 to 180 nanometers in diameter are located in the basal part of the cell, close to the cell membrane. Merkel discs are slow-adapting, tactile end organs consisting of Merkel cells which are in synapse-like contact with saucer-shaped terminal endings of myelinated A-beta nerve fibers. Final myelinated branches give rise to several unmyelinated branches, but sometimes Merkel cells at the marginal zone of the touch dome could be innervated by myelinated branches. Merkel cells transduce tactile stimuli into action potentials, which leads to a synaptic transmission. In turn, it drives A-beta nerve terminals to fire slowly adapting impulses. Merkel discs play a major role in the static discrimination of shapes, edges, and rough textures. It was also demonstrated that under some conditions, Merkel discs modulate the development of pain and itch. Hairy skin possesses very sensitive peritracheal nerve endings which detect hair deflection and light touch of the skin. Free nerve endings, Merkel discs, and the so-called pilar Ruffini corpuscles, which are similar to Ruffini corpuscles, are described in close association with hair follicles. Apart from this, hair follicles have specific sensory nerve endings which are called lanceolate or lanceolate nerve corpuscles. They form a collar of the hair shaft. The outer part of this collar contains nerve endings spiraling around the inner part, located parallel to the hair follicle and being in close opposition with hair follicle keratinocytes. This structure is compared to a comb of palisade. One dendrite can branch into up to 10 lanceolate nerve endings. Extracellular space between and around each lanceolate ending is filled with Schwann cell processes and collagen fibers. The whole lanceolate complex is located just below the level of the sebaceous glands and covered by a connective tissue capsule. Proprioception, literally the sense of self, in Latin proprius means own. Proprioceptors provide information about mechanical forces arising from the musculoskeletal system and include muscle spindles, Golgi tendon organs, and joint receptors. Muscle spindles are encapsulated nerve endings present in most skeletal muscles and distributed parallel to skeletal muscle fibers. They are especially numerous in muscles that require the fine movements, such as extraocular muscles, muscles of the hand and neck. Each muscle spindle consists of 4 to 8 specialized intrafusal muscle fibers. In Latin, fusum means spindle. Thus, force-producing skeletal muscle fibers are called extrafusal. There are 2 types of intrafusal fibers, namely nuclear bag fibers, 2 or 3 per muscle spindle, which in the central expanded part contain a dense cluster of nuclei, and nuclear chain fibers, which do not form an expansion and contain nuclei arranged in a chain. Intrafusal muscle fibers are striated as extrafusal and can contract except the central nuclear-bearing part. Myelinated type 1A sensory nerve fibers innervate muscle spindles, forming circular nerve endings in the central parts of both types of intrafusal fibers. These nerve endings are called annular spiral and are known as the primary sensory nerve endings of muscle spindles. Secondary innervation is provided by type 2 sensory nerve fibers that innervate the nuclear chain muscle fibers, forming nerve endings adjacent to their central part. These nerve endings are called flower spray. They may be spray-shaped or annular, spreading along intrafusal fibers. Most of the information concerning muscle spindle histology and physiology was obtained from observations in experimental animals. Studies on human muscle spindles corroborated these data. However, human secondary endings innervate both nuclear chain and nuclear bag muscle fibers, giving branches to the latter. Muscle spindles are unique among the sensory nerve endings because they possess their own afferent supply in addition to afferent innervation. This motor innervation is provided by myelinated type A gamma nerve fibers, up to a dozen per muscle spindle, which are called fusimotor nerve fibers. They form neuromuscular junctions at both poles of muscle spindle. Moreover, some larger myelinated type A beta nerve fibers, 1 or 2, also innervate muscle spindles, giving branches to extrafusal muscle fibers. They are called skeletofusimotor nerve fibers. The major function of muscle spindles is to send information to CNS about muscle length, that is, about the degree to which they are stretched. It is thought that the nerve endings of the type 2 nerve fibers detect the length of the muscle, and nerve endings of the type 1A nerve fibers detect the length and degree of change in muscle movement. Contraction of intrafusal muscle fibers under the commands of motor axons increases the stretch sensitivity of the sensory nerve fibers. The responses of muscle spindles to changes in length play an important role in regulating the contraction of muscles. For example, A-alpha motor neurons are activated via the stretch reflex, which resists muscle stretch. Golgi tendon organs are located at the junctions of muscles with tendons. Their connective tissue capsule is connected with a group of extrafusal muscle fibers at one end and with collagen fibers of the tendon at the other. Strands of collagen fibers, less compact than in the tendon lie inside the capsule. One or more myelinated type 1b nerve fibers perforate the capsule, lose myelin sheath, and form branches that intertwine with the collagen fibers. While muscle spindles detect changes in muscle length, Golgi tendon organs inform the CNS about changes in muscle tension. When the extrafusal muscle fibers contract under stimulation of their motor neurons, the muscle spindles are unloaded and silent, whereas Golgi tendon organs firing increases. Joint receptors gather information about limbs position and joint movements. 4 types of receptors are described in the joint capsule and ligaments, namely free nerve endings supplied by myelinated type A delta nerve fibers or unmyelinated type C nerve fibers, which are found in both ligaments and joint capsules. They are thought to respond to the extreme painful movements of the joints. Pacinian corpuscles and Ruffini corpuscles. Both types are found in the joint capsules and are innervated by myelinated type A beta nerve fibers. Pacinian corpuscles respond to movements of joints. Ruffini corpuscles respond to both movements and position. Nerve endings, which are similar to Golgi tendon organs. They are located in ligaments but not in the capsules and are innervated by myelinated type A alpha nerve fibers. They respond to joints position. Interoception is defined as the sense of the internal state of the body. As stated in Wikipedia, the contemporary definition of interoception is not synonymous with the term visceroception. Visceroception refers to the perception of bodily signals arising specifically from the viscera, the heart, lungs, stomach, and bladder, along with other internal organs in the trunk of the body. Interoception encompasses internal signaling more broadly, including, for example, blood vessels. Interoceptors detect pressure, stretch of the smooth muscles, noxious stimuli, chemical changes, osmolality, and temperature. Interoceptors are presented by free as well as encapsulated nerve endings, for example, Pacinian corpuscles. They are concentrated in the reflexogenous zones as well as diffusely distributed in various organs. Carotid sinus and carotid glomus are examples of reflexogenous zones. The carotid sinus is a dilatation at the origin of the internal carotid artery, in which there is a thinning of the tunica media, which is richly supplied by free nerve endings. They are baroreceptors reacting to changes in arterial pressure. The carotid body is located in the adventitia, in the bifurcation of the common carotid artery, and consists of cells and blood vessels covered by a connective tissue capsule. The cells are divided into 2 types, glomus type 1, or chief cells, and glomus type 2, or sustentacular cells. The carotid body is innervated by nerve branches from the sympathetic trunk and glossopharyngeal nerve. Glomus type 1 cells are associated with sensory endings of the glossopharyngeal nerve by synapse-like contacts. These cells are chemoreceptors that are primarily responsive to hypoxia and to a lesser degree to hypercapnia and acidosis. The low partial pressure of oxygen, the high partial pressure of carbon dioxide, and low pH in the arterial blood lead to the release of neurotransmitters, acetylcholine, ATP, and dopamine, into synapse cleft. The neurotransmitters bind to receptors on dendrite endings, triggering an action potential that relays the information to the CNS, where reflex changes in vasomotor activity and respiration are induced. Thank you for attention.